I'm Tina Mukherjee. I'm a faculty at the Institute of Stem Cell Science and Regenerative Medicine in Bangalore. And our laboratory tries to understand how blood cells form and develop. We use Drosophila as the model system. You must be intrigued if Drosophila, which is the fruit fly, has blood. And I'll tell you, yes, they have blood cells, which are very much like what the vertebrate or you and me have running in our body. They might be limited in what they can do, but yes, and our lab tries to understand how blood cells form and develop and what are their requirements. So our lab has actively worked on projects which have been aimed to investigate where metabolites come from, because what we are finding in our current studies is that metabolism plays an important role in cell fate determination. So as much as we understand metabolism for energy requirements, we eat food to derive energy, but our work in Drosophila has alluded to the fact that metabolism is more than just providing energy. It is actually supporting cell fate decisions and is a key determinant in how blood cells actually form. So the projects that we have worked under different funding agencies have been catered to identify what are these metabolites, what are the metabolic pathways from where these metabolites are being derived, and how do they actually govern cell fate decisions. So one of the projects that I have from the awarded to me was the Ramalinga Swami Fellowship. In that we were actually asking if there is any crosstalk between the blood cells or the progenitor cells and the brain, because it turns out that the brain has an important contribution in providing some important metabolites that are necessary for blood development. And again, if there are autonomous metabolites that are being generated by the blood cells within their own hematopoietic niche, this is another project that we worked with DST. It was an early career fellowship that was given to me. And with regards to CEFIPRA, we have that was the project that I also got awarded with, along with my collaborator, Professor Angela Giangrande at IGBMC in Strasbourg. So the idea in this project was to see, because blood cells don't come from one point in life. Blood cells are derived from different phases of development, which happen in the embryo, which happen later in the larvae or in the adult flies, which is comparable to how it happens also in vertebrates. You have a primitive wave of hematopoiesis that happens in the embryo and a definitive wave that happens later in the adult in the bone marrow. So these immune cells that are coming from different phases of their development, developing stages of the animal, what are the metabolic requirements that would shape up the immune cells in these different stages of development, is what the project with Professor Giangrande was aimed to identify. And the second part of the project was that if these metabolic needs that we find, or if we find that there are specific metabolic needs of immune cells in the embryo versus the larvae, if they are not met, what happens to the immune cells, how do they develop, and if they are contributing to any physiology in the animal which is independent of immune function, if they are perturbed or not. So the project that we have done is a collaborative project between our lab and the laboratory of Professor Angela Giangrande at IGBMC in Strasbourg. And while there are two parts to this whole effort or this exercise, one is the whole collaboration effort came together in the form of CEFIPRA that funded this collaboration. And second part is the value of collaborating itself with another lab. So I think there is a huge value in both these aspects. The level at which we have been able to conduct the research is really fantastic, because it has brought together two laboratories of complementary research expertise together to be able to solve a problem that would have not been achieved independently because Angela's lab focuses on has huge amount of expertise in transcriptomics, genome-wide analysis, in ChIP-seq, while our laboratory focuses on metabolic approaches to uncover blood development. So we both the labs are looking at blood development, but through different lenses. One is at the level of transcriptomics, proteomics, and our lab at the level of metabolomics. So this collaboration has allowed us to come together and bring all the platforms and get a holistic view of blood development, which otherwise would have not been possible. And this is the value of the collaborative initiative. And the best part is that our students from India side got the opportunity to be trained by the laboratory in France. Even though they did not get a chance to visit because of the COVID times, but they did have exciting interactions through meetings, online meetings, or through paper writing discussions. They were being trained in this collaborative atmosphere, getting feedback from labs outside our own atmosphere here, and I think that is very enriching. What CEFIPRA has allowed is to foster this kind of stimulating and in an interactive, enriching scientific environment. So I really applaud CEFIPRA because this is one of the very few international collaborative efforts that is really successful. It not only provides money for doing science, but also promotes exchange of students and visitors. Because most often we are doing science in our own laboratories, but the exposure that a student, not just for the PI, but more importantly for the student, that they get when they travel to another laboratory outside India and see the environment, interact with the students there, attend their courses, this is a way much more enriching experience. And what they bring back home when they come back to the lab is all the understanding, all the extra knowledge that they have been able to gain. And I think CEFIPRA has done a fantastic job in this regard. So with our project, essentially the goal of our project was to understand so blood development generally across systems, across different systems from humans to invertebrates, is a biphasic process where blood cells come from an earlier embryonic wave, which happens in the embryo, and that is called the primitive wave, and later the blood cells come again, which is the definitive wave, which happens in the adult in vertebrates, in the bone marrow. And in flies, it takes place in the larvae in a specialized organ called the lymph gland. So the idea of the project was to understand if developmental origin, so where blood cells come from, do they contribute, how does ontogeny define what their function can be? So if blood cells coming from the embryo have certain specialized function which are not seen in the blood cells coming from the later part of the hematopoietic waves. So if blood cells coming from the larvae are doing different functions. So if the idea is that are blood cells, all the blood cells same, or are they doing different functions? And if they are doing different functions, so is it important that where they come from define what they do? Now, this is essentially what the crux of the project was. And so what we have been able to identify, this is a fantastic effort in our collaboration, that what Angela's lab has been able to define is that through a series of transcriptomic analysis, which includes bulk analysis and at a single cell resolution, what their lab has been able to identify is that blood cells from different origins, be it from the embryo or from the larvae, are not the same. They have changes which tell us that the blood cells are of different, they have different qualities. So what her work says that, say the embryonic blood cells are slightly immature. They're metabolically also different from the blood cells which are there in the larvae. The idea that the larval blood cells are more mature and more metabolically more active is what we were able to identify. And this was very striking for us because we consider all blood cells to be more or less the same. Functionally, they're all going to be picking up and they're phagocytic cells, but what it told us that it's not the same. There are certain cells which are more phagocytic and the certain cells from the larvae are less, the larval blood cells are more phagocytic and metabolically more active, whereas the embryonic ones are not. So the point is now, are they functionally going to be the same? Because their states are not the same. So are they going to be functionally the same or not? This is where our lab contributed to and identified that blood cells contribute to various physiological functions independent of what we know of blood cells to be doing, that is fighting immunity. So as much as we understand blood cells from the standpoint of their immune requirements, what our work in India side has been able to identify are non-immune roles. And what we have done is that we have taken immune cells, manipulated their metabolic states, manipulated their functional proteins, the proteins that would define their different functions. That is the information that we got from the transcriptomic data. And what we identified is that the animals had developed conditions like metabolic disorders. So when the immune compartment was affected, the animals showed accumulation of lipids, so they were getting obese, they were much fatter. Also, there was another striking phenotype that we observed is that the animal growth was affected. So this is very striking. As I speak, I feel very I feel very excited because what we have been able to now show is that immune cells are contributing to how big or small an animal can be. So how tall or short you are can be defined from your immune compartment and how functional they are. So this level of understanding was missing in the field. We did not have any idea of growth regulation of an animal from the standpoint of an immune cells. We understand growth regulation from the thyroid, pituitary gland, and other organs, but we had no idea that immune cells can actually contribute to the scaling of the size of the animal. And this is what our work from the India side in collaboration with Angela's lab has been able to identify. The other part that we also have discovered is that as we manipulated the immune cells, the animals became fat or became deposited more lipids and also developed signs of insulin resistance. So which means that their sensitivity of an animal to how much insulin signaling can they see is also regulated by the immune compartment. So what we have been able to see is the development of metabolic disorders through the lens of an immune system. So we understand diabetes happens because of high sugar, there is high circulating sugar and there is insulin resistance. We understand people get fat or they become very, very acutely thin. But our understanding of this is from the standpoint of pancreas or the liver, the major metabolic organs. But what we are seeing in our results in Drosophila is that development of metabolic disorders can also have an immune intervention. So if immune cells are not kept properly, these animals have more susceptibility to developing metabolic disorders. So the idea of how physiology can be regulated through the lens of the immune system is what this whole collaborative effort has identified. Okay, so what we have been able to do is take this driver called hemolectin, which marks all blood cells in the Drosophila larvae. And what we did is that we ablated the blood cells. Essentially, we killed the blood cells. So that way the animals in this condition have no blood cells. And what then Sajjad and Anushri have done is that they have looked at the effects on the animal. What they have done is taken the tissues of these animals, which is essentially the fat body, brain, and smaller in sizes. So which suggested that which suggested that these animals' loss of blood cells led to a reduction in growth. So, which means that if you have an animal which doesn't have blood cells, you see a reduction in size of the animal. Now, the converse, if you ask, we also increased blood cells, which was doing PVR activated. So this is an activated form of a receptor. So what happens is here is that there are a lot more blood cells compared to the control. So now if we don't have blood cells, the animal is small, but if we increase the number of blood cells, what happens to the size? And what was surprising is that we found that these animals were slightly larger in sizes. So which meant that if you increase the number of blood cells and if they are functionally more active, they have a slightly, they can increase their sizes, whereas if you have blood cells when you have animals which have no blood cells, then the animal size is small.
I'm Nivedita. So I, along with Pierre and Rosie from Strasbourg, we have worked on the transcriptomic changes that are seen in immune cells from different developmental stages in Drosophila. So where Rosie and Pierre have worked on the transcriptome at a single cell resolution, focusing mainly on characterizing the different subpopulations, our team here has been focused on analyzing the metabolic changes that are seen across the developmental stages in chemocytes. So our analysis here actually has revealed that they are metabolically different. The hemocytes from different developmental stages, and many observations from our study has actually built a platform on which our team here has designed functional assays. And
hi, I'm Anushri, a postdoctoral fellow in TINA lab. So one of the core scientific contribution from the lab is when it was found that there is a non-immune functional immune cells in Drosophila in controlling animal growth, both in homeostasis, which is physiological condition, and in high sugar diet, which is a form of nutrient stress. So it was at this point it became interesting to understand that how do these immune cells themselves behave in such kind of nutrient stress scenario. And it was very interesting to observe that like here you can see that we observed there was change in the morphology of these immune cells, and interestingly there was change in the metabolic status of these immune cells. And this is very interesting because we already talk about metabolic heterogeneity in these Drosophila immune cells and their impact on functional responses. So currently I'm trying one of the focus area is try to develop genetic tools to understand or to perturb these heterogeneous population of immune cells and their functional roles.
India and France friendship.